So most of us here know that the microbiome is really important for human health and disease. I probably don't have to convince most of you of that. We know that the microbiome has been implicated in a number of different health and disease conditions that range from colorectal cancer to inflammatory bowel disease and even cystic fibrosis. And we also know that the composition of the microbiome is really complex and that it's affected by numerous factors like host genetics, diet, medicine use, and the environment that you're in. So given the important role that the microbiome plays in human health and disease, we're interested in exploring patterns and variation of microbiome composition on a large scale in the hopes that this would help us better understand just how the microbiome is affecting our own health and disease states. And to do this on a large scale, we would need a lot of data. And fortunately, there are hundreds of thousands of microbiome samples that are already publicly available via online repositories like the NCBI's Sequence Read Archive. Now, previously, our lab analyzed just the metadata of around 400,000 human microbiome samples from multiple body sites from NCBI Sequence Read Archive. Now, this plot on the right shows the number of samples that were available over about a decade and where in the world those samples come from. Right away, we can see that the vast majority of microbiome samples come from Europe and North America, indicating that we have a pretty big overrepresentation of like a westernized, industrialized microbiome in publicly available data. Now, despite this imbalance in sampling, we're still really excited at the prospect of leveraging this wealth of data that's already available and using it to create one unified database. So to that end, we integrated samples using a single pipeline. Now, we started by taking over 245,000 16S sequencing human gut microbiome samples from the NCBI SRA. These samples came from over 460 projects, so we know they're a very diverse sample set. They come from people young and old and sick and healthy all over the globe. And we put these samples through a uniform processing pipeline. Along the way, we excluded studies that had very few samples, as well as samples that either failed some of the processing steps or didn't pass quality control. And this resulted in a final database of over 168,000 samples. Now, this is, to our knowledge, the largest currently available data set of its type. And we're really excited about this data set as a resource for everyone within the microbiome community, and not just us. So to that end, the Human Microbiome Compendium, as we refer to this data set, is already freely available at our website at microbiomemap.org. And you can also load the data through our R package of the same name. Now, because we wanted this to be a resource for everyone, we wanted to make sure the data set had as little of our fingerprint on it as possible. So we were pretty lenient when filtering samples to create this data set. But for our own analysis, we did slightly filter it further to exclude samples that had a lot of unidentified taxa or had very low read counts. And once we had this data set, which most of the analyses that I'm sharing today will be using this filtered data set, once we had it, one of the first things we wanted to do was to see where in the world these samples were coming from. So we classified the world into 8 distinct geographic regions. These were defined by the UN Sustainable Development Goals. And right away, you can see that the vast majority of our samples come from Europe and Northern America. We have over 90,000 samples from this region alone. And this is in pretty stark contrast with some of the other world regions, like Latin America and the Caribbean, which we have about 1,200 samples from, and Northern Africa and Western Asia that we have about 1,400 samples from. And you might have noticed that I said there are 8 world regions, but there are only 7 shown on this bar chart. Now that's because there's a small region in black close to Australia. This is called Oceania, and we had only 4 samples total from this region. So they're excluded from further analysis just because we wouldn't be able to find any real patterns in only 4 samples. But I bring that up just to highlight how uneven microbiome sampling has been historically, while it has improved over recent years. Now, once we had broken our samples down into the world region that they came from, we wanted to know if there were differences in microbiome composition between these regions. And we thought that there might be because we know that things that directly affect the microbiome, like host genetics, diet, and medicine use, are to some extent reflected in geography. So we used principal coordinates analysis to get just a broad overview of sample composition and how it might vary. This plot shows every single sample within the compendium. Each point is a single sample, and it's colored by the world region that that sample came from. Now, you might be able to see some patterns or a little bit of clustering here, but it's pretty difficult to tell what's going on because there are so many samples that these points are all overlapping with each other. And so I wanted to break this analysis down even further and show just each region by itself. Now, these plots are a little bit different because instead of having each point be a single sample, a point is representative of the number of samples in that area on the plot. So a lighter color indicates a higher sampling density, so like a more common microbiome for that region, whereas a darker color is a lower sample density. And when we look at the plots for these two regions, Europe and Northern America in the top right and Central and Southern Asia in the bottom left, right away we can see differences. It looks like the most common microbiome for Europe and Northern America lives somewhere around the origin, whereas for Central and Southern Asia, it's a little bit to the right of the origin. And the spread of the samples in these two regions is also pretty different. The samples from Europe and Northern America take up pretty much the entire breadth of the plot, which is not the case for the samples of Central and Southern Asia. And when we plot the rest of the regions, we see even more stark differences. So this was really exciting because it just demonstrates that there are compositional differences within microbiomes from these different world regions, as we were hoping to see. And once we knew that, we wanted to zoom in even further and look at specific taxa that might be driving these compositional differences. Now, we used a differential abundance analysis to do this. So we took 65 of the most abundant genera within the compendium, and then we tested them for differential abundance between each pair of world regions. This plot is just a broad overview of that analysis, and it shows the number of taxa out of the 65 tested that were differentially abundant between each pair of world regions. So you can see that, for example, 53 taxa were differentially abundant between Central and Southern Asia and Latin America and the Caribbean. Every single one of these 65 taxa tested were found to be differentially abundant between at least one pair of regions. And you can see that the majority of these regions have a lot of differences. So I wanted to zoom in even further and pick out just a few taxa that are highly abundant and significantly differentially abundant. The first taxon is Bacteroides. Now, this was one of the most abundant and prevalent taxa within the compendium. And historically, Bacteroides has been associated with a westernized and industrialized microbiome. This plot shows the distribution of the relative abundance of Bacteroides in every sample within a region. So a peak towards the right of the plot would mean that there is a lot of samples with a high amount of Bacteroides in that region, whereas a peak towards the left of the plot means there are a lot of samples with a low amount of Bacteroides in that region. And right away we can see that, like we would expect, there are a lot of samples from Europe and Northern America in pink that have a lot of Bacteroides. But interestingly, it looks like almost all of the regions have a lot of samples with a lot of Bacteroides. In fact, the lone outlier here looks to be Central and Southern Asia in light blue, which is the only region that doesn't appear to have a strong concentration of samples with a lot of Bacteroides. The next taxon that I wanted to highlight is Faecalibacterium, which in contrast with Bacteroides has historically been associated with a non-westernized and non-industrialized microbiome. And we can see that the distribution of samples here looks a little bit different. So while there are still a significant number of samples in Europe and Northern America that have a lot of Faecalibacterium, there are almost an equal number that have very low amounts of Faecalibacterium. And we also see really high relative abundance of Faecalibacterium in Sub-Saharan Africa, Northern Africa and Western Asia, and Latin America and the Caribbean. And the last taxon that I want to talk about is Prevotella. So this one is also been associated with a less westernized and less industrialized microbiome. And this is perhaps the most stark contrast we'll see today, because we have very few samples from Europe and Northern America with a high amount of Prevotella. Most of those Europe and Northern America samples have very low amounts of Prevotella. In contrast, the samples from Sub-Saharan Africa, which are in dark orange, appear to have a lot of Prevotella in general. And then because this analysis has really shown us that there's a lot of compositional differences between these world regions. And I think that highlights the need to continually get more diverse microbiome samples to better understand what's going on worldwide. Because we think that with such low sampling in some of these places, we might be missing some of the taxa we'd be able to find otherwise. And so to get at that idea, we used a rarefaction analysis where we took a random subsample of microbiomes from a given region, and then we counted the number of unique taxa in those microbiomes and repeated this analysis for increasing sample sizes in every region. And right away, we can see that we have way more samples from Europe and Northern America than anywhere else, but also that the line for Europe and Northern America looks to be leveling off pretty quickly, indicating that we're not really finding a ton of new taxa, even though we have tens of thousands more samples to assay. And this point is underscored if you look at the inset plot, which shows the same data, but on the y-axis, it's the number of taxa per million reads. By the time we assay all of the samples from Europe and Northern America, we're finding 0.39 taxa per million reads. Whereas with some of the other regions, we're still rapidly finding new taxa, even as we run out of samples to assay. And this is something I just wanted to highlight because I think further sampling efforts would probably bear more fruit and we'd be able to find a lot more interesting and novel things if we looked at microbiomes that we haven't studied so deeply before. And very quickly, I want to thank everyone in both of my labs, the Bleckman Lab at the University of Chicago and the Albert Lab at the University of Minnesota, as well as both of my advisors, Ron Bleckman and Frank Albert, and specifically a few members of the Bleckman Lab who contributed to this compendium project. Rich Abdil started this project when he was a graduate student in the Bleckman Lab, and it's safe to say that work would be nowhere near where it is without him. Everyone highlighted contributed to this work in some way, and I'd also like to thank our collaborators, specifically Casey Green and Sean Davis, who have been working on this project with us since the beginning. I really appreciate everyone's input, and it wouldn't be where it is without them. And if we have time, I'm happy to take any questions, but I know we're running short.
Thank you. One quick question, yes.
Hi, thank you very much for a nice presentation. I was wondering whether you looked at the functional level, how unique or like how different the populations were from each other?
We haven't looked on a functional level. One new analysis that I found really interesting that I wasn't able to share today is we did like a phylogenetic gain analysis. So the database that I've been using for this was not phylogeny based, but we added in another database, a reference database that was phylogeny based. And I think that was pretty consistent with what we would expected. Regions with like higher alpha diversity in general, you find more phylogenetic gain when you add in samples from that region. But we haven't done too much with function yet.